Welcome to part 2 of the neuromuscular basis of movement. In this segment we will be looking at how peripheral nerves develop or arise or emerge out of the spinal cord, so the shift between the central to the peripheral nervous system. We'll look at action potentials, which is a description of how an impulse is generated and travels down the neuron. And then finally we'll look at synapses, which is how the impulse travels from one neuron to either another neuron or onto the target tissue. So this diagram is a cross-section of the spinal cord. And what you're seeing are three levels of the spinal cord. And let me just orient you a little bit. So here's our cross-section. And here is kind of this butterfly-shaped or H-shaped segment here, and this is the gray matter in the spinal cord, and that's going to be where we see a lot of the cell bodies. And then all this stuff around here is the white matter, which are the ascending and descending tracts that go up and down the spinal cord itself. And what we're going to look at again is how we transition from the spinal cord out into the peripheral nerves. And so ultimately we want to look at how each segmental or spinal peripheral nerve is developed, and then how that combines to create named nerves. So for example, when we look at the quadriceps, that's innervated by the femoral nerve. Well, that femoral nerve has to arise out of different levels of the spinal cord, and so that's what we're going to figure out, is how that all happens. Let me point out some areas that you need to know, or some terms you need to know. So first of all, just to get oriented again, this is the dorsal or the back part of the spinal cord, and here facing you is the front or the anterior or ventral part. So when we look here, a spinal nerve comes out of every level of the vertebra all along the spinal column, and so that spinal nerve is made up of both fibers coming from on the dorsal side, and that's carrying just sensory information, and then also areas from the ventral or anterior side, and that's carrying motor information. And then they combine to make a spinal nerve that has many neurons and carries both motor and sensory information. So these spinal nerves are what we talked about in the first video that were named like C6 or T2 or L1. They're given the first letter of whatever segment of the spinal cord they're coming from, and then the number of whatever level. So let's look at a little bit different view. This is a superior view, and just to make sure again you know what you're looking at, this is the vertebral body, and this is the posterior arch with the spinous process. So this is the back, and this is the front of one spinal segment in the cervical area. And again, here's the cross section of the spinal cord with the characteristic little dark or gray matter outlined here, and then of course surrounded by white matter. So that is within, the spinal cord runs within the central canal or the spinal canal of the vertebra. And then, as I mentioned, at each vertebral level you have one spinal nerve that comes out, and that's what you're seeing here is this spinal nerve. So this, let's just say this is, C5. And again, we have, if we look way into the spinal cord here, we have some neurons that are coming out from the dorsal side. That's going to carry sensory information. Some neurons coming out from the ventral side, that's going to carry motor information, and then they're going to blend into a spinal neuron here. So here's the names, the spinal neuron coming right here. This is a little simpler diagram, and it can be a nice one to look at. This is one of those pictures that if you understand everything on this page, on this slide, and you can explain it, then you're going to really understand the information that's important. So again, it's not colored in, but this is a cross section of the spinal cord, so this is the dark matter, and this is the white matter. Here's the dorsal side. This is called the dorsal root. So if you think about a root kind of coming out, so that dorsal root is carrying only sensory information. Here's the ventral root coming out of the anterior side, that's carrying only motor neurons, so only motor information. Then again they blend together, and here's the spinal nerve, here's the term, here's the spinal nerve. So that's carrying both motor and sensory information. So we have a few more things we need to look at now. So this little expansion here is a ganglion. Ganglion just means a bubble or a balloon or a big circular area. And so inside this ganglion are the the cell bodies of the sensory neurons. So you may have to go back and look at a slide from the previous video where we looked at the anatomy of a sensory neuron. And if you recall, the cell body was kind of in the middle. There was a big axon coming out to one side that came from the periphery, and then a little bit of an axon going into the spinal cord. And the cell body was kind of in the middle. So that's what this is. The cell bodies are all in the middle here. The rest of the axon is coming in, making up the dorsal root, and then a whole bunch of long axon is coming here, coming all the way out, eventually innervating the skin or some other structure away from the spinal cord. So that's the dorsal root ganglion. So again, the spinal nerve here has both motor and sensory information. So now let's go past that a little bit, and the spinal nerve splits. It splits into an anterior or a ventral, and then a posterior or a dorsal ramus. So the word ramus means branch. So the spinal nerve branches off dorsally and ventrally. So we have a dorsal ramus and a ventral ramus. And I know it's really close to the word root, but root and ramus don't mean the same thing. So the root of something would be close to the center, and then of course the branches would be further away. So branch and ramus. You'll see here there's a pretty big difference in size between the dorsal and the ventral ramus. So the dorsal ramus is going around, and it's going to innervate the muscles and the skin of the back. And then the ventral ramus is going to come and innervate either the muscles and skin or of the anterior trunk, or it's going to go to the extremities. So let's say that this is a spinal nerve in the cervical area. So that ventral ramus is ultimately going to go and innervate the upper extremities. So that's why it's so much bigger, because there are many, many more neurons that have to go innervate the upper extremity than that have to go innervate just a little bit of space behind on the back of the neck, for example. Alright, so I think maybe we should do this one more time, just so that you're really clear, and we'll use the next slide, which is a little more colorful, but exactly the same thing. So now we're in the lumbar area, and we just know that because of the size and shape of the vertebra, so don't worry about that. But again, anterior, here's our vertebral body, posterior, here is our spinous process, and then what you're looking at here are essentially the tenderloins. Or the back extensor muscles. So let's go back in here. Here's our cross-section of our spinal cord. That's probably looking pretty familiar to you by now. Here's our dorsal root, and here's our ventral root. So those are carrying, again, just sensory information in the dorsal root, just motor information in the ventral root. Here's the dorsal root ganglion. That's got the cell bodies of the sensory neurons. Here's our spinal nerve. Both motor and sensory axons or neurons are making up that, are gathered in there. Here we get to the point where we're branching off. So here's the dorsal root. Here, let me go on this side where it's actually labeled. Here's the dorsal ramus branching off, and that's going to come and innervate the muscles of the back, and then some that are going to go innervate the skin, because remember, there's both motor and sensory axons in there. And then the ventral ramus is branching off here. That's going to go innervate, in this case, since we're in the lumbar area, now it's going to go innervate muscles and skin of the lower extremity. So far, if you're following along on your lecture outline, we're on page 3 and we've only gotten to 3 items. We've gotten to emergence of the dorsal and ventral roots, the dorsal rami, and the ventral rami. And we're going to look at 2 more slides that explain this, but the point is to understand how the nervous system transitions from central nervous system, which is spinal cord, to peripheral nervous system, which are these spinal neurons or spinal nerves exiting at the spinal cord level. So very different way to look at this. Again, this should look familiar to you, this type of structure here, because it looks very much like what we looked at for muscles and for connective tissue. So we have a nerve. This is our spinal nerve right here, and it's made up of, here's our myelinated and unmyelinated neurons. So each one of these is a nerve cell or a neuron. They're bundled into little fascicles, and then many little bundles make up a spinal nerve. So one nerve has many, many neurons in it. And if we look back here, again, there's our spinal cord. We know the dorsal roots. Here we go. Coming into the dorsal side of the spinal cord, that's going to carry just what kind of information? Sensory or motor? Right, sensory. Here's our dorsal root ganglion. What's in the dorsal root ganglion? Good. Cell bodies of the sensory axon and sensory neurons. Ventral root then has motor information, and again you can see all these. So all these lots of axons are coming together, and you're seeing all these axons in one spinal nerve root. And again, this looks like a cervical level, could be any level, it doesn't matter, but this could be C3, or this could be T2, or this could be L5. This is the spinal nerve coming out of one spinal level. One more slide. Again, just a little color-coded. It's showing you the direction of transmission, and it's color-coded, so sensory information basically is coming into the spinal cord, into the dorsal horn of the spinal cord, and then motor information is coming out of the spinal cord from the ventral horn, anterior horn, and coming out. And again, all those axons, though, are traveling in one spinal nerve. So I know that can be complicated and a little difficult to understand. Again, let me go back to this slide here. This is a really good slide, I think. It's a little more simplistic. And if you can draw, label, understand, explain this slide, then you can feel comfortable that you understand the anatomy. All right, we're going to move on to a related but a little bit different topic called dermatome. And a dermatome is an area of skin that's innervated by just the sensory component of a single spinal nerve. So remember, we have 31 paired spinal nerves coming out at cervical, thoracic, lumbar, and sacral levels. And so this is a map of all the dermatomes in the whole body. But each dermatome, there's a dermatome associated with each spinal nerve. So you can see here that in the trunk, let's take the trunk first. Basically, if you can picture, use your x-ray eyes, look through and see the spine and the vertebral column, and you can picture the dorsal and ventral rami coming out, and their sensory portions are going to wrap around and just innervate these segments of the trunk. So here's T1, T2, T3, T4. So they just basically come out of the vertebra and just wrap right around the trunk and innervate the skin. And so you can map that pretty easily. Now in the upper extremities it looks a little bit different. It's all stretched out, but they're still fairly straight or fairly parallel. And then the same in the lower extremities, but except now they're kind of angled. So what you have to remember is that when we think way back to embryological development, when we're first developing as an embryo, remember the limb buds are way up here. And so the limbs, so if you picture this guy with his arms, shoulders abducted at 90 degrees, well then the dermatomes in the arms would look very much like those in the trunk, just nice and horizontal. If we pictured abducting the leg to 90 degrees, we would see that. But we'd also have to add some external rotation, because as the limb bud starts here in an embryo and grows out, it also internally rotates, and so that's where the swirly pattern comes. But again, if you abducted the hip and externally rotated it, then the dermatome would be nice and horizontal like it is in the trunk. So basically, again, using your x-ray eyes, you see the vertebra. Out of that vertebral column, you have spinal nerves coming. They divide into what you can see here is the ventral side, and that would be where the ventral rami are coming to innervate the skin of the anterior part of the trunk and the extremities. And then the dorsal rami would go to innervate the skin of the back. Some of the landmarks that we use, the T4 tends to be right about the nipple level, T10 about the umbilical level, and then in the genitalia area is where the sacral nerves innervate. Another important concept when we're looking at the spinal nerves exiting the body is how we go from having a spinal nerve, say C5, to actually having a nerve with a name like the musculocutaneous nerve. And so that's where the concept of a plexus comes in. And the word plexus just means network. So remember, anatomists weren't really trying to be fancy, they just used the names that made sense to them. And I have to say that this is a nice slide, but if you look at it, it is not really named correctly. This is not the brachial plexus, this is the cervical plexus. So we have 3 plexuses that we're going to look at: the cervical, brachial, and lumbosacral plexus. So this is really the cervical plexus, and it's made up of nerve roots coming out from C1 through C4. I know C5 is on here, we're going to put that in with the brachial plexus. So C1 through C4. So again, we just looked at how those spinal nerves are created. Now we're taking those spinal nerves, and what happens is they combine in complicated ways with axons from the spinal nerves at different levels. And they combine in magical ways, and then again we end up with named nerves. So we're not going to pay too much attention to the nerves that come out from the cervical level. So let's go to the brachial plexus, C5 through T1. So here are the spinal nerves. They're coming out, C5, 6, 7, 8, and T1. And you see them here on the left side as well. And they combine, so the roots, the spinal nerve roots are what we just looked at. They come out at each level. That's in purple. They combine into trunks, and then they split, those trunks split into anterior and posterior divisions. You will be thrilled to know that you are not going to have to diagram the brachial plexus, you're not going to have to know. You can just say the spinal nerve roots of the individual level spinal nerves come out and combine, and a miracle occurs, and then we end up with nerves that have names. So the axillary nerve, the radial nerve, the musculocutaneous nerve, median nerve, and ulnar nerve. So if we look at the musculocutaneous nerve, this is the nerve that innervates the biceps. So if we trace that back, we can see it's getting elements from C5, from C6, and from C7 here. And that's making up the musculocutaneous nerve. So every nerve with a name, not every nerve, most nerves with names are really made up originally of axons from a variety of spinal levels. So that's going to be important as we think about pathologies later on. At this stage of the game, I just want you to know how it happens. But again, you don't have to know the magic of, does which anterior and posterior divisions create what, and which trunks are important. You don't have to do all that. You just have to understand the nerve root comes out individually at each spinal level. It combines with other nerve roots, and then what you get after that combination occurs are nerves with names. So when we start to study the different regions of the body in detail, so let's think in the upper extremity here when we go to study the elbow and forearm, and we're going to study the biceps. you're going to have to know all the attachments of the biceps, all its actions, and then what you're going to have to know is that it's innervated by the musculocutaneous nerve. You aren't going to have to know the musculocutaneous nerve is made up of these different spinal levels. I'm not going to ask you to memorize that, I just want you to understand it. And then, as I mentioned, we have a umbosacral plexus, and that's what you're seeing here, and this is made up of nerve roots. I know it says L4 here, it actually starts just a little bit higher at L2 down through S4. So again, the roots, the spinal nerve roots, are in purple. So we have an individual spinal nerve at L4, L5, S1, etc. They again combine in magical ways in anterior and posterior divisions, and what we get after all that combination are nerves with names, like the sciatic nerve, for example. And look at how big the sciatic nerve is. That gets combinations from virtually every spinal level. So you have an assignment where I've asked you to find out, to look up and find out what spinal levels make up different nerves. And you can find that information in the Kendall muscle testing book. You can find it in, I believe, your kinesiology book where it talks about the muscles. So there are a lot of different ways, but you are going to have to look that up to find out which plexus and then again which spinal nerve roots make up the individual named nerves. But it's not something I'm going to have you memorize. I'm having you go through that on your assignment just so that you understand how it works. Okay, enough about that. We're going to shift gears a little bit, and we are going to talk about now how nerve impulses are generated and travel down a neuron. So how do we actually get a nerve impulse to go from coming out of the spinal cord in that spinal nerve root, to the muscle? Or how do we get it to go from your fingertips, up into your spinal cord, up to your brain to find out that you touched something hot? So what we're talking about essentially is excitation of a nerve or a neuron. And this is very much an electronic event or an electrical event, because what we're talking about are movements of ions, which are charged elements within the body. And most of these slides have a little more detail than you need. A lot of them will talk about the sodium channels and potassium channels and things like that, and that's more detail than you really need. I think it's helpful to just, again, remember sodium and potassium ions are charged, and it's the flow of these charged particles in and out of the neuron that actually generates the impulse. But again, I'm not going to have you go into that level of detail. So let's take a look at the terms here. We have, let's just talk about action potential. So potential here means, is an electrical term, meaning a difference in positive and negative charge. So if there's no difference, then that potential would be zero. But generally there's a difference, and what we're talking about, we mean from between the inside and the outside of a neuron. So let's look at the graph here. Here's our membrane potential in millivolts, and you can see here that our resting potential, when nothing is happening, the neuron is at rest, there's a negative 70 millivolt difference between the inside and the outside of the neuron membrane, so the nerve cell. So inside the nerve cell, it's negative 70 millivolts versus if the outside is 0. So there's a difference. And that negative 70 here is, it's not an absolute number. That can range anywhere from negative 60 to negative 90 millivolts. So they just thought negative 70 was a good place to put it. This graph is always from the standpoint of inside the neuron, because it's all about the neuron in this case. The next term here is, so that was resting potential. So at rest, when nothing's happening to the neuron, the value inside the cell is negative 70 millivolts. So in order to get an impulse to travel down, we have to get ions to flow in and out. And basically, positive ions end up flowing into the neuron. So that depolarizes the neuron. So polarization, like if somebody is polar opposites from you, that means they're very different. If we depolarize it, we make them more similar. So you can see that as positive ions flow into the neuron starts becoming depolarized, less negative. And if we get to negative 55 millivolts, then all heck breaks loose, and there's very rapid ion flow into the neuron. And so that causes this rapid spike where the neuron goes from, on its inside, from being negative to being positive. It doesn't matter how high. How high depends on a lot of other factors that we won't worry about. But so this creates this is the action potential, this whole spiky thing here. So this is showing you that there's lots of neurons flowing in, and then, so that's called depolarization. And again, at the peak, we have a lot of positive neurons inside. Then, and that, that process occurs in response to some stimulus, which is not illustrated here, but there's a stimulus that causes those channels to open. So, for example, if you touch something hot, the stimulus was the hot input to your sensory receptors in your skin. They caused sodium channels to open, and the neuron started sending that sensation of hot temperature up along your axons, into your spinal cord, up to your brain, so that you eventually became aware that you were touching something hot. So once that stimulus is removed, then the positive ions get pumped back out, and so inside the cell it starts getting more and more negative. That's called repolarization. We're becoming polarized again. And this cellular machinery kind of usually overcompensates, and so it pumps a little bit more out, and so it becomes hyperpolarized. So it pumps a little bit too much out, but then it equilibrates and you end up back at resting potential. So during this repolarization phase, especially here, we actually become hyperpolarized, and so this is a refractory period, which means resting period. And during this period, it would be either very difficult or in some cases impossible for another action potential to occur. So it gives the neuron a little bit of a rest. Once it's back to resting potential, then it could have another impulse. So we're going to look at a variety of pictures to help give you different examples and help you really understand what we're talking about. So this is a very similar picture, and again, a little bit more detail than you need. You don't need to know about sodium and potassium and all that, but we're going to see this same setup here on our graph. We're going to always have membrane potential on the vertical axis, and the horizontal axis is time. Of course, this happens very, very quickly. So let's look at really from the standpoint of the neuron. So here's our membrane. This is a neuron at rest, so it's polarized. It's negative on the inside, positive on the outside. And again, what's the number? How different are they? Right, it's between negative 60 and negative 90 millivolts difference on the inside. Then, as we start to depolarize and the positive ions are flowing in, that kind of progresses down along the axon. And so really you can follow the progression of the nerve impulse as the ions flow in, and so this would correlate with that spike starting to go up on the neuron. And so the depolarization continues down the neuron, but behind it now the neuron starts to get repolarized, where it's becoming back more negative again. So let's look at one more picture of that. Nope, this is it. So this is just showing you the flow of polarization or depolarization down. So again, let's look at this one. So depolarization is this, where the positive ions are flowing in, and that depolarization flows down or along the axon. And then repolarization is when it's becoming more negative again. The positive ions are getting pumped out. And again, you can see that happening here. Positive ions getting pumped out, and that's kind of following along behind. So if you think about an impulse traveling along, much like an electrical current travels along a nerve, that's really very close to what is actually happening. Alright, so what happens then when we get to the end of the neuron is we have to decide if we can continue this impulse. So that's when we get to talking about a synapse. And remember that the end of the neuron, where the axon ends, it doesn't actually touch another structure. There's a little bit of a gap. And what this slide is helping to show is that there could be a lot of different ways that information either coming into or going out of a neuron. So there can be synapses between, so this is the cell body, this is the axon, and here's this little triangle here is the end of the axon. This is a dendrite, and so you could have, if you're trying to send that sensory impulse along a sensory nerve, and then you want to send it along another sensory nerve to get it up to the brain, for example, you could synapse onto another dendrite, then that would send it to the cell body along the axon, and then again you could send it somewhere else. So you could synapse between an axon and a dendrite. That's an axodendritic synapse. You could have an axon synapsing right onto another axon, axoaxonic, or you could have an axon synapsing onto a cell body, axosomatic. We want and we would like to think of the nervous system as an impulse traveling from A to B, then from B to C, then from C to D, but it's a lot more, which is what it would look like if this was what our nervous system looked like. If we had, again, our cell body, our axon, we have our impulse going along, here's our synapse, goes on to the next item, and it keeps going. And it would be nice if it was this simple. Let me point out a few more terms before we move off of this one. Since, if we look back here, since we have a lot of different options for synapses, in order for us to just talk about a synapse in a generic way, instead of using the terms axon and dendrite, we're going to use the term presynaptic element. So the portion, whether this is an axon or a dendrite or whatever, the presynaptic element before the synapse, and then we're going to just talk about the postsynaptic element, because it could be a body, it could be an axon, it could be a dendrite. So presynaptic and postsynaptic elements, and then of course the synapse here. But what I was saying before is it would be really nice if everything was just one straight line, but many, many synapses are occurring simultaneously. So this picture is a lot more representative. This is an actual electron microscope. So this big kind of tannish, grayish looking thing in the middle is a cell body, a soma. These are all the synapses coming onto that one cell body. So each cell body is getting multiple inputs. These these neurons could be going on to, these presynaptic neurons could not only be coming onto this body, but simultaneously onto other cell bodies or other axons or dendrites. So there's lots of simultaneous synapses going on all the time. And I think that's what makes neurology so exciting and interesting, but also so complicated and challenging to understand. However, we're going to step back, thankfully, and just simplify it and look at what's happening generically at one synapse. And you've seen some similar pictures to this when we looked at how muscle contracts. But we've got an impulse coming down, the presynaptic element, whatever that is. And remember, since we don't touch, we have what's called a synaptic cleft. There's space between the pre and the postsynaptic elements. And so in order for the impulse to cross, we need to use some neurotransmitters. So along this presynaptic element would be what we saw on this slide. This depolarization flowing down along that element. Once it gets to the end where those neurotransmitters are located, the neurotransmitters then that are in these little vesicles or bubbles migrate toward the end of the synapse, presynaptic element, and they release the neurotransmitters. Those chemicals flow across the synaptic cleft, and they have to bind onto specialized receptors onto the postsynaptic element. If enough bind, then the impulse may be continued along the postsynaptic element. So there has to be adequate neurotransmitter, there has to be adequate binding, and again, we'll look at what happens when in future classes when that doesn't go so well. But that is basically the sequence of a synaptic transfer. So we'll look at one more picture here. And remember I said it may continue. So the other little wrinkle here is that some synapses are what we call excitatory, where they do allow the impulse to continue. Some are inhibitory, where they stop the impulse. So let's look at this. So there's two synapses here, right? Here's this neuron. This is the inhibitory neuron, and this is the presynaptic element. So this is the main one right now. So right now, just kind of ignore this gray one over here. Right now we have depolarization occurring, traveling down this presynaptic element. Neurotransmitters are getting released, they're flowing across, they're binding, and once adequate or sufficient numbers travel across and bind, then the impulse will continue in the postsynaptic element. Well and good. Let's look at this situation over here. We had an impulse traveling along, but we also have this neuron that's coming in and synapsing, and if this inhibitory synapse goes, it shuts down what's happening on this neuron. Ooh, ah. So this is where that idea of multiple inputs comes in that makes things a little complicated. So I can give you an easy example of this that I'm sure you've all experienced. So let's say that this is pain traveling in this neuron because you just whacked your shin on the coffee table. So pain is traveling along and you are definitely getting the signal of pain. Now what you do is you reach your hand down and you rub your hand on your shin where you whacked it, and all of a sudden you aren't feeling quite as much pain, and that's because the rubbing has been acting as an inhibition or an inhibitory synapse. So the rubbing sensory input has diminished the pain sensory input that came from whacking your shin on the coffee table. All right, I think that's enough for this segment. So we will pause, and then you're going to want to go on. I don't know, this one's a little out of sequence. I'm sorry about that. Let me just show you these. I apologize for these being out of sequence. This again obviously is action potential, and it gives some more slides about action potential in the progression. So you can take a look at those as well in relationship to the progression of the impulse. Now I promise, this is it. So you can give your brains a rest before you go on to part 3. Thank you very much.